Today we will discuss DNA not as biologists, but as future litigators, judges, and legal scholars. DNA is the most powerful biological identifier currently used in courts worldwide. Unlike fingerprints, which identify patterns, DNA identifies a person at the molecular level. When properly collected, preserved, and analyzed, it can link a suspect to a crime scene, exclude the innocent, establish paternity, or identify disaster victims. But its strength in court depends on understanding what it can and cannot prove. DNA exists inside the nucleus of almost every cell in the human body. As shown in this slide, the nucleus is the command center of the cell. For legal purposes, this means that almost any biological material, blood, saliva, semen, hair roots, skin cells, all can potentially yield DNA. This is why even touch DNA can be used in criminal cases. However, the smaller the sample, the greater the risk of contamination, degradation, or interpretive error. Inside the nucleus, DNA is packaged into chromosomes. Humans have 23 pairs, 22 autosomes, and one pair of sex chromosomes. Each chromosome has a short arm labeled P and the long arm labeled Q. Why is this legally relevant? Because DNA profiling focuses on specific chromosomal regions called loci. These loci are standardized internationally for forensic comparison. When experts testify, they refer to allele matches at these loci. DNA is composed of nucleotides, each made of a phosphate group, a sugar, and a nitrogenous base. The four bases are adenine, thymine, cytosine, and guanine. The sequence of these bases forms the genetic code. For legal analysis, we are not examining medical traits. We are examining repeating sequences that vary among individuals. This slide compares DNA and RNA. DNA contains deoxyribose sugar and thymine. RNA contains ribose sugar and uracil. In forensic law, we focus on DNA because it is stable and durable. RNA is more fragile and rarely used for identification. DNA's chemical stability is one reason it is admissible decades after a crime, provided chain of custody is intact. DNA forms a double helix. Adenine pairs with thymine, guanine pairs with cytosine. This complementary pairing allows replication. In court, replication matters because forensic laboratories amplify small DNA samples using PCR. Without this base pairing mechanism, forensic identification would not be possible. This karyotype shows the 23 chromosome pairs, autosomes, and sex chromosomes. In forensic practice, sex determination is often performed using the AMEL marker. This is useful in sexual assault cases or unidentified remains. However, sex determination alone does not individualize a person. It is only one data point. Here, we see chromosomal loci, specific positions on chromosomes. DNA profiling focuses on short tandem repeats or STRs at these loci. These are repeating sequences that differ in number between individuals. The number of repeats is what creates individual variation. Courts rely on these differences to calculate match probabilities. Genes contain exons and introns. Exons code for proteins, introns do not. Importantly, forensic DNA profiling examines non-coding regions, primarily introns or intergenic DNA. This means forensic DNA does not reveal medical conditions or traits. It is used strictly for identification. This distinction is important when addressing privacy concerns in court. Only about 1.5% of the human genome codes for proteins. Approximately 98% is non-coding. Forensic profiling targets these non-coding areas. Why? Because variation is high there, making them ideal identifiers. And because they are non-coding, ethical objections about revealing health information are minimized. Restriction enzymes act like molecular scissors, cutting DNA into fragments. Historically, this was used in RFLP analysis. Today, we primarily use PCR-based STR analysis. But understanding restriction digestion helps explain early DNA fingerprinting cases and challenges to methodology in court. PCR or polymerase chain reaction is the engine of modern forensic DNA analysis. It amplifies tiny amounts of DNA through cycles of denaturation, annealing, and extension. Even a few cells can generate millions of copies. However, PCR is extremely sensitive. That sensitivity is both strength and weakness. It can detect minimal evidence but also contain contamination. In litigation, contamination and laboratory protocol compliance are frequent grounds for cross-examination. In the fictional case shown in Table 1, Suspect A is excluded as the source of the crime scene sample. Suspect B, on the other hand, matches the crime scene sample at all 13 STRs. A calculation of the frequency of Suspect B's genotype based upon the STR allele frequencies within Suspect B's ethnic group reveals that the likelihood that a random member of this ethnic group has this profile is about 1 in 1.5 billion. It is important to understand that this number is the probability of seeing this DNA profile if the crime scene evidence did not come from the suspect but from some other person. To regard the number as the probability that the suspect is the source of the crime scene evidence is to commit the prosecutor's fallacy. This is the logical fallacy of equating the probability that an animal has 4 legs if it is an elephant. So it's high. With the probability that an animal is an elephant if it has 4 legs is low. To go from one probability to another requires the use of Bayes' theorem and some prior and close in parentheses before the DNA profile, close parentheses, probabilities of the suspect being the source of the evidence. In addition, it is important to note that the probability of 1 in 1.5 billion is substantially increased if the actual source is a person related to the suspect, especially a sibling. 1 to 2 mismatches may still allow inclusion, especially if mixtures or mutations are involved. 3 or more mismatches generally indicate exclusion. 4 to 5 mismatches confirm exclusion. To conclude, DNA is the gold standard of biological identification. It is powerful, statistically robust, and globally standardized. Yet, it requires strict adherence to collection protocols, chain of custody, laboratory validation, and proper statistical interpretation. As future lawyers, your role is not to memorize nucleotide chemistry, but to question methodology, probability, contamination risk, mixture interpretation, and the limits of inference. DNA is evidence, not verdict. This closes the presentation.